Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. My goal is to find the exceptional people in their fields and, bring them to you, the listener, and ask them good questions and get information out of them that maybe typical magazine articles and interviews wouldn't get, the good stuff. Today I have Joanna Mazet, PhD. She's a professor of epidemiology and disease ecology at the UC Davis School of Veterinary Medicine and executive director of the UC Davis One Health Institute. So she works on global health problem solving for emerging infectious diseases and conservation challenges, which is the emerging infectious disease part is particularly appropriate right now with this COVID nightmare. So, Joanna, thanks for coming. How are you doing?
I'm doing well. Thanks for the honor of having me.
Yeah, and I guess since you work with veterinary issues, and since this is, I guess, what they call supposedly a zoonotic disease, a virus that jumped from a bat or a pangolin or something into people, your background's pretty appropriate, right?
Yeah, I mean, for the last decade, I've been leading an international consortium of amazing people working all over the world to find viruses just like the SARS CoV-2 that's caused this terrible tragedy that we're living in now. And so during that time, we really feel proud that we've helped the countries where we were working, more than 35 countries, over the last 10 years. And we've discovered a lot of virus, including more than 100 coronaviruses.
So in particular with this coronavirus, have you been able to look at the sequence? Have you been able to work on it specifically, or is your work still just in general on these kind of viruses?
Well, as far as the in general part, we've published studies showing all sorts of sides of coronavirus diversity through our findings on PREDICT. But on this one specifically, we've certainly looked at the sequence. We've been helping the laboratories in the countries where we're working to identify cases in their countries. In fact, before the sequence was even available, we used our project that I mentioned, PREDICT's platform, which is a broad-based platform to help the countries surrounding China rule in and rule out introduced cases before the specific tests could be developed, because earlier even. So just in a few days, it might that be.
So what aspects of all this are you studying, or what are you involved in helping to create or figure out?
Sure. Well, like I said, one of the big things that we've been doing for the last decade is to understand the viruses that are out there and able to spill over from animal hosts into people and cause epidemics, and hopefully never again pandemics, and raise the flag that we need to be ready for disease X that could come from any of these spillover events. So we've been working on that. We have a lot more people listening and interested now, unfortunately, because of the tragedy. I wish that we had done even more to prepare before this one happened. But I think we're super lucky that this virus is not more deadly than it is, because we can see how easily it spreads. And the only thing that is really saving us right now is our behavior of staying home, and that the virus isn't more deadly. So I know we're all very stressed and living with this tragedy, but I hope that we will take heed now and jump into action and really prepare, so that if another and when another dangerous pathogen spills over, we're more ready.
Well, how are we supposed to prepare? What are we supposed to do?
Okay, yeah, sure. So right now, I'm working with the National Academies of Science, Engineering, and Medicine to look at all of the aspects of controlling this current outbreak, but also to look more forward thinking and to find virus and understand the characteristics by which they transmit, how they spill over, who's at most risk. And we can do that in a couple of different ways. We certainly have proven through the PREDICT project that we can go to the least resourced countries in the world and work with them to get out in the field, train their people to be super safe, identify the high-risk transmission zones or interfaces where people are interacting with animals in a particularly stressful way for the animals to cause this spillover event to happen. And then we can get into the laboratories and we can use broad platforms to identify viruses. And that's super useful. So the training and the identification platforms means that those countries are more prepared right at the start to find what's happening in their countries and stop it. It also allows us to start this catalog or database that's not just the virologic or sequence information about those viruses that are out there, but it includes information about all the different ways that the virus can infect people. And it also includes looking at how one virus may be more risky than another. It's only by going out and discovering about 500,000 viruses that we believe are out there and ready to spill over into people that we can start to understand which ones are likely to be the more dangerous ones and which ones aren't, and begin to preemptively target our mitigation measures like staging, preparedness, equipment, and even having better pipelines for rapid vaccine development.
Well, how are you going to go out and characterize these viruses? Do you pick animals that people are likely to come in contact with or unlikely to come in contact with? Do you profile their virome?
Yeah. So initially, as we've been working in primarily Southeast Asia and Africa, initially what we do is we go talk with the folks, especially the ministries of environment, ministries of health, and ministries of agriculture, and bring them all together. And we say, where are you seeing fevers of unknown origin in people and seeing the specific drivers, risk factors for these spillover events? Those include changing landscapes, increasing human populations, and biodiversity. So we know that people have access and are being exposed, but also that there's fever going undiagnosed that we should be looking into. Then we sample the animals and the people in those interfaces. In the what we call the proof of concept now in PREDICT, we only sampled really intensively non-human primates, bats, and rodents, because all of our mathematical modeling showed us that those were the targets that were the most likely to have viruses that could spill over to us, and also the animals that we're interacting with in ways that put us at risk. What we believe we need to do now is take that even a step further and say, okay, we can look at the most risky. That's how far our money's gone so far. But we need to look actually at all mammals and water birds that are really the hosts of a lot of things that are available to spill over to us in all the interfaces that are left in the world. And we're calling that the Global Virome Project, which will take countries coming together, people, projects, teams coming together and working in a collaborative way to identify these risk factors, the hosts, the geography, and the viruses themselves.
It just seems so expansive. I don't know how would you pick it? I mean, are there, bats supposedly seem to harbor a lot of really terrible viruses, but maybe more commonplace creatures under stress of, places like being in wet markets or, loss of habitat, that's more likely to cause problems. I mean, how do you pick and how do you know what will work and what won't? What if, I don't know, if animals that we're in common contact with for some reason end up becoming an intermediate carrier of something, and then because we're in contact with them a lot, cats or dogs, that it gets to us.
Okay, so historically, we have, as a human race, been exposed to things that our own microbiome that sometimes make us sick because we get stressed or other things. And we've been exposed over history to the domestic animals that we live with as pets and we eat. And so we find less and less emerging infectious diseases from those species because we are less naive to them and their microbiome. So what we're really concerned about now are the viruses that can spill over from wildlife hosts into us or into those domestic animals that we live in such close contact with. And we would then be the naive host, as would often the domestic animals. And so we can be more susceptible to severe disease because we haven't seen those viruses in evolutionary history. So you're right, it is extremely expansive. What we're talking about doing, we're talking about looking at all of them, all of the mammals and water birds, and we know that it can be done, and we know how much it would cost. It's whether or not people are willing to jump in and do that. We really believe that we should understand them as much about viruses as we do about bacteria, and we have all the tools to do that now. The cost is it's in the low billions, but before this terrible COVID tragedy, we knew that just finding all of this virus and all of the circumstances would cost just 10% of what we had spent on previous large epidemics like SARS. And now we know that it's decimal places lower than that when you consider a pandemic of this scale. So really finding the viruses is the first step to preparing for them, and it's much, much less expensive than responding to even a portion of a large epidemic.
Yeah, it makes sense. How do you know, again, like what about bacteriophages that would become, virus-like to us?
Sure.
Is that a potential vector? I mean, how do you find all these viruses, and then are you sequencing them, or how do you know what they do based on their sequence?
Yeah. And you can't know everything based on their sequence. You have to do additional tests, but first and foremost, we need to understand the circumstances they're in and find the virus, because if it's not likely to come into contact with humans, well, we're less worried about that than one that comes into contact with humans frequently, right, and has more opportunity to spill over. There are things we can do with the sequence, and we can reverse engineer and make a sort of safe model for working with the virus so that we can understand more how it uses its receptors and binds to human cells and other animal cells. So that's all part of the follow-on work that needs to happen. But yes, we do sequence these viruses. First, we use sort of what we call a more conservative approach, in that we aren't interested in every viral species that's already known to man. We're interested in finding the ones that are known to cause problems, but more importantly right now, ones like SARS CoV-2 that are unknown but might cause problems. We need to find the virus, identify it, sequence it to identify it, but then we also need to find the host, because when we find a virus that is infecting multiple hosts, and especially affecting multiple hosts that are really different from each other evolutionarily, like a mouse and a tiger, then we know that that virus is more likely to be able to jump into humans as well. So it's not just the viral characteristics. That's something people have focused on for decades and decades, and it's important work. But we also need to understand the transmission potential and what human behaviors and what we're doing to the planet are driving the risk for spillover.
Yeah, and then, I mean, from what I understand, there's plenty of viruses that just are persistent in us and in other animals, and then we have endogenized viral DNA. So from what I've also read, sometimes endogenized viral DNA can be activated, in times of stress. So how do you evaluate in a chimpanzee, let's say, what's endogenized that could be reactivated? What's persistent in the chimpanzees that's not harmful to them, but maybe when they're stressed, now all of a sudden it activates and, is harmful to someone else? I mean, how do you characterize what to look for?
Right. So we we did an extensive look at the literature and identified about 40 risk factors for spillover and then going human spread. And then we asked all of the researchers that wanted to participate around the world that were experts in this field to rank the likelihood that these different risk factors that had been identified here and there, or we identified through the PREDICT project, which ones of those should be given the most weight, which were most important for spillover. So it's imperfect system in that we haven't studied viruses enough to understand all of these things that you're asking, and we absolutely can and need to do that. But we need to start finding them to even be able to do that. So we have to start somewhere, and we can start now. But when we worked with those researchers all over the world, 70 different researchers helped to rank the risk factors. We started to basically almost crowdsource the science around the world in a collaborative way to put together a platform where people can enter their own viral findings and share them. At the same time, use the risk platform to identify where they might rank as far as spillover potential. And as new findings are coming out, we can refine that system together.
That's great. So what are the risk factors of, from most risky to least? I know you can't name them all, but what are some of the ones that surprised you?
Okay, well, ones that didn't surprise me were, RNA-based viruses, because they tend to mutate faster and transmit a little bit easier. So definitely there were things about the viruses themselves that have been known for a while that are definitely in there. Ones that surprised us more, even though we've been working on this, I guess it wasn't a recent surprise, but over time, this host plasticity seems to be way more important. And by that, I mean that host range, how many hosts that can infect and how different those hosts are, seem to be much more important than, for example, being most genetically related or a near neighbor to another virus. So, for example, if looking at the first SARS coronavirus, finding the most closely related SARS coronavirus wasn't the thing that made that coronavirus most likely to spill over, because we've found a lot of those now. It was something else. And it's that something else that we need to narrow down. But this host switching seems to be one of the biggest factors that's giving us a clue to that right now. And that probably lies in the spike protein that people are hearing a lot about, or that key that fits into the receptor on susceptible host cells like humans.
But what would make a virus able to infect many different types of hosts? Is it just that it just happens to target a very common entry path into cells that a lot of creatures share?
That's one thing for sure. Some viruses, they can jump easily because they can hide better from the host immune system, or they replicate better in the host because they can hide from the host immune system. So we're looking at those pathways as well. The immune pathways in the host themselves give us clues. So we can look at the viral characteristics like the spike protein for coronaviruses, and then look at the ACE2 receptor, which is the receptor in the host cells like in humans. That seems to be how the virus enters. And then once it enters into the cell, we can look at how the host immune system reacts. So hosts that react less severely tend to be better amplifying hosts than hosts that react really severely, because the ones that react very severely tend to get sick and maybe die before they transmit the virus too much. So all of these things are really important, but it's sort of differs from virus to virus. So it's not simple, but we're starting by this collaborative effort. We call it the One Health effort, where we are more holistically looking at how the health of humans, animals, and the environment, including plants and our foods, are really interconnected, and that we have to look at all of those together. We can't simply be looking at only one pathway at a time. So that takes experts in different fields coming together to make this happen.
So what are some of the near-term goals of PREDICT and of the project?
So PREDICT right now is finishing up, but we are currently assisting in the 28 countries that we were most active in towards the end of this decade-long project. We're currently assisting the amazing professionals in those countries to have what they need in their laboratories to do more exhaustive testing for COVID disease through finding SARS coronavirus 2. So we're really supplying the technical assistance to the labs to help them get ready, especially in countries where they don't have widespread community transmission. And we are also putting our folks that, again, those amazing scientists and health practitioners who were trained through the PREDICT project in this One Health approach, we're putting them, allowing them, enabling them with salary support that they didn't necessarily have before to contribute to their government platforms. So we're seeing our PREDICT scientists, some of whom were the first people to ever discover or write a paper about coronaviruses in their countries. They're becoming the experts that are sitting on the national task forces in their countries and helping them do the infection prevention and control. So that's most important right now because we really need to halt this pandemic. In addition, we are going back through all of our archives and testing species like pangolins. Lots of people are hearing about pangolins that maybe weren't our target hosts in PREDICT, because again, we were looking at the bats, primates, and rodents. But now we want to understand this particular SARS CoV-2 even better. So we're going back to our archives and looking to find the different hosts who could be infected by this virus and understand the transmission and spillover risk, because there's still work to be done to find out exactly how this virus did spill over and keep it from happening again, and figure out clues from this specific dangerous coronavirus that will help us understand the other coronaviruses that we have found and that can be found in the future.
What do you think is going to happen with the current, SARS CoV-2 pandemic? It doesn't seem like it's stoppable at all. I mean, do you think it's just we're going to have to just accept that it's going to be a persistent virus that comes around every year, or is just here until it affects a very large percentage of the world's population?
Yeah, I mean, it's difficult to say. I think that right now, just stressing that we do need to stay at home, give our healthcare systems a chance to be up and running. We need to, work really hard on therapies and on vaccines. I mean, I would hope to say that we won't need the vaccine far into the future, but I think out of an abundance of caution, we're going to need to be vaccinating and being careful about this coronavirus and others. So we're really right now in a situation where we are staying at home to slow spread, to protect the systems that will protect us, like the healthcare systems. Once we all start going back to work and leaving our homes, we're going to unfortunately see some upticks in cases and illness. And though it remains to be seen how we'll handle that, but it's likely that we're going to need some phased approach to getting back to work that probably doesn't include everybody going back all at once, so that we can continue to sort of stall this pandemic while we get vaccine available. There's, of course, always the hope that seasonal changes in either our immune systems or what's happening in the environment will change the transmission patterns of coronaviruses, but the evidence is not there to say that will happen. So that's a hope, not a plan. Out of an abundance of caution, again, we need to give the systems the chance to get ready and get busy on the vaccines. And I'm super excited and pleased about how fast, for example, the CEPI coalition has come into play to get vaccine trials going. But it remains to be seen how long it will take to get manufacturing going. So yeah, we can get through this. We can get through this one. And I think we should turn our energies to being prepared for the next one. And you asked me that question before, and I don't feel like I got to the place where I really wanted to say, we can prepare our systems as far as our health systems to be more nimble than was proven to be a problem in this current situation. We can provide the infrastructure and the planning to be able to handle any of these viruses, as long as we're ready to trust science and to jump in quickly at the beginning and not stall. So if we work together and we allow our system to not be so siloed disease by disease, but include planning for the unexpected, we can be ready for the next one. We can have testing systems and pipelines ready. We can have government organizations working together instead of against each other. And we can really change the way we think and react to the warning signals that we get from other countries, but also just from our own communities and environment. And that's really what's going to fix this for us.
Yeah, I mean, the next time something like this happens, I mean, there's no, even though if we have a healthcare system, it really doesn't seem like it can do anything. It puts people on ventilators, let's say, but they die anyway.
Right.
So
right.
It just doesn't seem like there's no solution unless we have a vaccine or an antiviral. So it seems like, I guess,
Right.
like what you're doing is identifying potential threats, but we would need to be on a very fast track to find antivirals and or vaccines in order to really have an effect.
But if you find the viruses ahead of time, and you can look at the ones that are making animals sick or in tissue culture, infecting cells and causing cell death, you can start to identify what we don't know now because we haven't studied viruses enough. You can start to identify what characteristics of which viruses we need to target vaccines to and which characteristics we don't. If we don't start finding those viruses, I agree with you, it is kind of hopeless. We'll just be sitting here waiting for the next one. And that's what we've been talking about for the last 10 years. Let's stop doing that. Let's stop chasing the last one and then throwing all of our money at that last one. Let's get prepared for the future. Let's have those that deeper understanding of the viral communities and diversity, and then start to understand what makes certain viruses dangerous and other ones not. And we haven't done that work yet. It is possible though. And I don't want you to have like a kind of defeatist message here. We are seeing the vaccine, antiviral, all those pipelines are being dramatically improved. They were to some extent before this, but there's been a major sea change. So if we can find a silver lining out of this whole terrible tragedy, it is that that people are thinking differently about this now. And hopefully, because we have this basic call to action, hopefully we won't revert to our sort of human nature of saying, well, that'll never happen again, or that's in the past, and this is more important what's happening here today. We need to be ready, and we have the technology and the momentum to do that.
Yeah, it makes sense. So what's the best way for people to see some of the findings, some of the commonalities that, that Pareto of the risk factors? Is that published, and where? How do people find out more?
Yeah, so there's a lot of published literature. If folks wanted to go to predict.global, www.predict.global, you can actually look at the maps of everywhere we've done surveillance, and even click on the findings. You can see the test results of the viruses we found and in what species. And if you're a scientist and want to link out to GenBank and take a look at those viruses, you can do that as well. You can also see the publications that have been coming out of the project are all listed there. That we're probably, because of this current situation, we might be a little behind on posting some of those, but we're getting those up. We have more than 200 scientific publications that have come out of the project that you can check out. And I definitely think this host plasticity piece would be something good to look at. The first author is Dr. Christine Johnson, and I'm the senior author, last author on that paper. And then there's our paper in Science on the Global Virome Project and why it should be done. So there's a lot of opportunity to review those materials. You can also look at globalviromeproject.org for more information.
Okay, well, that's great. It's a lot of resources.
Yeah.
Well, Jonna, thank you for doing what you do and paying attention to this, because as we all know now, it's very important, far more important than we realized. So thank you very much.
Yeah. Well, thanks for shining a light on it and doing what you do to get there.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.